All right, so the first thing that we need to make sure you understand is what your group has to, make sure you scan this thing, what your product has to look like. You have groups, you know what you have to do. The second thing that we have to do is go over the concepts covered in the lab. This lab covers a lot of concepts, but we're only going to touch on a few of them, and then we'll keep developing them over the rest of the quarter. In order to help you understand what's going on here, and you're going to see a lot of vocabulary words in this lab that you may not be aware of. There are two methodologies, two strategies that we could use for you to learn this material. I could lecture you about every concept that's in this lab, like osmosis. Let me just go ahead and write the big ones down that you'll see again. Osmosis. Solute, solvent, solution, ions, transport, across membranes. If you're coming in late, you have to go get a pass from the office and sign in. So these are kind of the big ones that this lab touches on. All right. You could also talk about plant development. Plant reproductive organs. Cross-pollination. Ladies, go get a pass. Oh, you got. All right, put it here and sign in. Cross-pollination. Gametes, fertilization, and the list goes on, but those are kind of the big ones that this lab could touch on if you really wanted to focus in on. What we're going to be talking about today a little bit is solvent, solute, and solution, and next week we'll talk about ions, and then after that we'll talk about transport, osmosis, crossing the membrane, what is a membrane, biomolecules, etc. So there's a lot to discuss, and we need to unpack that slowly. So rather than starting with this and spending a week lecturing on and going through problems on all of this, I thought we would start with a lab, let you experiment with a fairly simple concept of control groups. These are the main ideas of the lab, right? Controls, experimental group, control group, and also to experience this, to experience all that. And take a look at it without really understanding the vocabulary. So as you're making your observations during this lab, as you're planting the seeds, if you're coming in late, I need you to get a pass. No pass, it's just, you need you to scan in and explain why. You need you to scan in anyways and explain why. All right. So You experiment, you have controls, experimental group, control group. What is an experiment? So that's really the key to what we're doing and why we're doing it right now. We're going to talk today a little bit of solute solvent solution because it directly applies to how you're setting up the lab. All of this directly applies, but it would be too much, I think, to discuss all this now. But we will be talking about it. So as you're making observations in your notebook during the next week, because you'll be taking notes every time you come in to water the plants, you're going to make notes of what you see and how you're measuring it, etc. Take a look at what's happening and think about it, because you will be seeing it again. We will be discussing and identifying what is happening at each stage of this plant development, of this experiment. So this is kind of what we call, have you ever heard of discovery learning? No? No, sorry. So this is called discovery learning. So it's better, some of us believe that it's better that you discover what it means for a plant to germinate, that you discover what pollen is, that you rather than me telling you, it's better that you discover it. So it's experiencing these hands-on labs, I feel, is the best way to introduce major concepts. Sometimes we'll do the opposite. Sometimes I'll explain a lot of different concepts, and then you'll do the lab, and you'll see if you can make that concept work and see that concept in action. For instance, when we cover osmosis, it will be our next lab. When we look at osmosis, you're actually going to make a gel, a polymer of a gel, and it's called agarose gel. It's a seaweed protein that we're going to make, and you're going to add acid to it and an indicator called phenolphthalein, and you're going to put it in a base, and you're going to see the osmosis, the movement of water from high to low, happen in a cube. And you're going to measure the rate of that movement of water. So it's really interesting. It's a very interesting lab. It's colorful, it's vibrant, it's action-packed, if you will. No explosions. But those will come later. We'll do, not an explosion, but we'll release the energy in sugar all at once, and you'll see how much energy is in sugar. It's an amazing experiment, but we'll have to do it at the end of the year. Yeah. Experience, yeah. Sorry, it's very messy. So the whole point is, during this lab, you're going to experience all of this, but we're not going to identify it. You're not going to be tested on transport across the membrane, on plant development, on the parts of a flower. You should look at the parts of a flower. You should think about this being a compound flower and how this flower is actually made of 1,000 little flowers. Like, that's really some really cool and interesting concepts that you can really take a look at. But I'm not going to be testing you on that. What's our focus? Because you guys keep telling me you want to know what's important here. You want to know what's the purpose, right? And these are good reasons. These are good questions. So the main purpose here is to look at controls, experimental groups, control groups, and see how an experiment is designed, and see if you can actually answer a question using an experiment. That's the whole point of this lab. It's an actual experiment. It's a lab, but it only becomes an experiment when you have what? Controls, experimental group, and control groups. Once you have this, now you got an experiment. Otherwise, it's just a lab, which I shouldn't say just a lab, because labs are cool too. So labs can be experiments, but not all labs are experiments. So an experiment is when you're dealing with collecting data, and you have a control group, an experimental group, and all that. And then you have to analyze it, and we'll go through the steps next week. But the bottom line is, you're going to experience all of this. You're going to focus on this. And then, as I said, don't forget when you're looking at it that these three, and we'll talk about it before you leave today, these three are important, okay? because you can't do it unless you understand what concentration is. Another word that I should add to this list is concentration. And you're going to be looking at concentration a lot this year. Concentration is a big deal. So how concentrated something is a big deal. So, and it actually, this idea of concentration, solute, solvent, solution is going to feed into osmosis and diffusion. It's going to feed into how a cell works. Think about, when you're thinking about concentration, we talk about solute, solvent, solution. Think about being a doctor. When we talk about dose, what is dose? What does the word dose mean? And so what really are we worried about when we say dosage? Concentration. Because you give someone too strong a dose, you'll kill them. You don't give them a strong enough dose, it's not going to help them. So as a physician, dosing the drug is very important, whether you're a veterinarian, a nurse, or doctor. So concentration of a solute in a solution is incredibly important. What solution would you be worried about in your body? Or any animal would be worried about in their body. What solution do you think a doctor's constantly worried about? You don't have to get a pass because I guess there's a lot of people put skin in. Okay, explain why you're late. Yeah? Blood. So when you're talking about the concentration of blood, well, how much sugar's in your blood, you're worried about that if you're diabetic. So the concentration of sugar is important if you're diabetic. I'm diabetic, so I know this. It could be, if you're anemic, it could be the concentration of iron in your blood or concentration of oxygen in your blood. So these concentrations become very important when you're talking about surgeons, nurses, anesthesiologists, assistants, physician assistants. If you don't understand this concept, it's going to be hard for you to understand much of anything in biology. So when you're playing around with this experiment, you're discovering new ideas and maybe seeing things that you haven't seen before. Consider focusing on these things that I'm highlighting now. But for today, for most of today, what I'd like you to do is work in your groups and come up with, number one, what materials do you need? A materials list. Two, you're probably going to have to do two first. The second thing that I need from you is the procedures. Remember, the procedure you have in the lab is not the one we're going to use. We have this equipment. They took it in the back. They're filling up the little pots with soil for you. So the soil will... I feel like we should try to save you as much time for tomorrow as we can. And we're going to have the pots filled with soil for you. We're going to have the salt solutions already set for you. Unless you want a different salt solution, then we can talk about it, all right? But you want your procedures. You want to have your data. What data are you going to collect? What is your hypothesis? This is not in order. You understand that, right? Because you have to have a hypothesis first. What's your question? This actually looks like it's almost... What question are you trying to answer with this experiment? Okay. What's your procedure? What data are you going to collect? And the last thing you guys have to work on is answering the questions, in every question that you can that's in the lab. The lab has a lot of questions. Answer every question that you can. Now, I'm going to let you work for most of the period. If you tell me that you'd rather keep working as a group, and we can cover solvent solution, solute solvent solution tomorrow while the lab is running, we can do that, all right? So whatever you feel you're at as a group, if you're being productive as a group, I'm going to leave you alone, and we'll talk tomorrow about the content. If you feel like you're done, you got everything done, and most of you are done, then we'll go ahead and move into explaining the content now. Does that feel good? Do you understand what's going on? How you feeling? If you're feeling like you need things to slow down, you feel like you need things to speed up, you feel like you need to give me that feedback. All right? It doesn't have to be here in this open session. You might be embarrassed about it, but you can come and talk to me after class. If you ever feel like, some kids feel like I'm picking on them, like teachers pick on them. If you feel like I'm too harsh, not harsh enough, if I'm, you can give me that feedback. I'm not promising I'm going to do anything about it because I might disagree with you, but I will promise that I will listen to you. All right? So you can tell me anything. I've been told everything from F you to, I'm an old man, right? I've dealt with a lot of stuff, so you don't have to worry about if you feel like any kind of certain way, then we need to have a discussion. I think I've been clear on what needs to happen. I think I've been clear on what's not acceptable. If I haven't been clear on what's not acceptable, I'll repeat it again later. All right, are we good? All right, have a good day. So I just want to make sure we're clear on page. Can someone give me the salt lab? It's right behind you, yeah. As you flip through it, the first 2 pages are for teachers. You shouldn't have got it, but I photocopied the wrong version. You have the introduction. Read the introduction. It really has a lot of information. Yeah? You forgot one? There's one on the front table. Oh, I thought you said you forgot one. Yeah, of course you can use the restroom. Don't forget to scan out, take the pass, all that. So on page 8-6, you have materials, and then you have a procedure. Remember, that procedure for A is not what you're going to be doing. If you want an outline for that procedure, we went over it yesterday. I posted it online on YouTube last night. This morning, I posted it to Schoology and to your web page. But the outline for the procedure is already done for you. We did it yesterday in class. You should have notes on that. Option B is only if you want to do it, so I'm not even going to we're not even going to discuss option B since it won't even start till next week. And now on page 8-9, it says salty solution. It says, what date were the seeds planted? It asks you for a date. Obviously your date's going to be tomorrow. It asks you for the salt solution used. Salt solution used. It asks you, and it gives you a percent. What percent salt solution? And then it gives you a table, and in table A, it asks you to fill out, what is the amount of soil you're using, and then it asks the question of the type of soil, and then it says the type of water. And then it says the amount of water. And then it says the number of seeds. Now you're going to plant more than one seed in a container, just so you know. So the way this was set up originally this lab was set up for that every group in this room would have one pot with one set of seeds in it. And you'd have one salt solution, then we would collect, class data on it. The only problem I have with that is that then some of you would never see anything grow, correct? Because some of you would have a solution that's going to kill the plant. So some of these solutions are going to kill the plants, so you're never going to see anything grow. Others will allow the plant to grow and it will thrive. Which solution is going to do that? Well, we don't know. If you only had one solution, some of you would never see it grow. So I don't like that. So the way we set it up, and it took a lot of effort. And again, my assistants have already created your planters, your 5 planters. They already have soil in them. They're in the back. They're already in their trays, in the light tables. They're ready to go. We're going to count out the seeds. They're making the salt solution. So they're going to start mixing in salt. So we'll have that. So that being said, what you're going to need is this information for all 5 of your groups. You have your control group, where you would answer the question. Obviously, you'd have the date. The salt solution used would be 0% salt, right? Is that correct? The amount of soil is going to be the same. I don't remember what it is. I'll give it to you tomorrow. The type of soil, everybody's going to have the same type and same amount for all the groups. So this is all going to be the same for all the groups. Does that make sense? For the control group and for each of the experimental groups, you're going to have all the same. So type is garden soil. Now, to be clear, that you would normally not say garden soil. What you normally say is loamy, silty, sandy. You would use words like acidic, basic, neutral. But we're not going to do that. We're going to use the word garden soil for all of them. The amount, let's just say, I'm going to just give it a number, 50 milliliters. And that's going to be the same for all of them. You have a question? There's a question.
Are do we have the option to use magician mix or like the 50-50 mix?
We're not going to use either one. You're going to plant with the soil's already been put in. It's going to be garden soil, and you're going to put. Well, let's just go on and answer this. The type of water, type H2O. Here it's going to be distilled water, DI water, distilled water, DI equals distilled. All right, so distilled water. The rest, all the rest of these are salt water. Salt water, salt, salt, salt. A number of seeds. The number of seeds I haven't figured out yet, but it's probably going to be around 5 seeds each. So everybody's going to get about 5 seeds. There might be a difference between groups. So again, you're going to have this little pot. You're going to have in the pot. You have this little pot. And in the pot, you're going to have soil. And on the soil, you're going to put this little seed. There's probably going to be 5 of these little seeds in here. All right. And you want to keep them on the surface. Keep the seeds on the surface, because what's going to stimulate their growth, what's going to start them growing, is light. If you bury them too deep, they won't grow. They won't start growing. So try not to bury them too deep. So that's what and of course each one of these is going to be, this is going to be 0.5%, this is going to be 1%, this is going to be 2.5%, and this is going to be 5% solution. And each one of them is going to have a different bottle. We have this big jug. It's going to have really concentrated salt. It's going to be like 20% salt. And from there, we will take and make the 5 smaller bottles, right? There's going to be 5% and et cetera, all the way down, okay? And then we'll keep diluting them until you're going to have water for each of these. And every day, you're going to add a certain milliliter of water. The amount of water, by the way, which I did not put here, the amount of water is going to be about 10 milliliters And that's going to be across the board, 10 milliliters. See, the amount of water, the amount of soil, the quality of the soil, the amount of light is all going to be the same, because that's how we do science. We keep all the variables the same, except for the independent variable. We want to change that, and we want to see how the dependent variable changes because of the independent variable. So that's how we work science. So that's your job. You're gonna have to. I just pretty much did a lot of that for you, because this page, page 8-9 was confusing to a lot of people. So I want to make sure you don't. So 8-9, 8-10, this covers that, and you guys can fill out table C. You'll be filling it out tomorrow. Then there's some questions you have to discuss. You may not be able to answer till after tomorrow. We will be answering questions together. This is not a test. This is a learning exercise. Is that correct? Is that understood? So I don't need you to get me the right answer. I need you to think about the questions and then ask questions tomorrow during our discussion phase. Let's discuss it. Discussion means discuss, not me lecturing you. So I want you to ask me questions. I want you to, if you don't understand, I want you to ask for clarification. I want I want a discussion, not you staring off into space, right? And then just writing down the stuff that I write on the board and walking home and say, I understand. Yeah. That's not a discussion. All right. So if you understood all that and you're ready to go, you guys have a job to do. Write your procedures out. Make sure you have your data table set. Make sure you know what you're going to do tomorrow. And tomorrow you're just going to come in and do it. Is that clear? Okay. Oh, and you're going to come in, by the way, just so you know, you're going to have to water these plants every day for 5 days. Over the weekend, they'll have water. All right, I'll make sure of that. But tomorrow, so you're going to come in tomorrow. Wait, do you guys have lab tomorrow or Friday? Tomorrow. Yeah, tomorrow. So come in and do the lab tomorrow. But then Friday, you have to come in and water it in the morning. One or more of you have to come in Friday morning to water your plants. On Saturday, Sunday, I'll make sure they're watered. Then Monday, you have to come in and water the plants. Yeah. Someone came back. Yes, you may. Don't forget to scan out. All right, we're all good. Any questions? So since you have no questions, we're all good. You're working as a team. All right, get your stuff ready then. You got the rest of the period. Unless, if we all get done, if we are all done before the end of the period, I can start going over the content. Okay? Huh? I can't.